Primary polydipsia and diabetes insipidus, characterized by the production of large amounts of dilute urine with or without thirst. Urine-specific gravity is less than 1.006. In both primary polydipsia and diabetes insipidus, the urine-specific gravity is typically less than 1.006. This is due to the dilution of urine caused by excessive water intake or impaired concentration of urine. In primary polydipsia, individuals consume abnormally large amounts of water, leading to the kidneys excreting a large volume of dilute urine. This excessive water intake overwhelms the kidney's ability to concentrate urine effectively, resulting in a low urine-specific gravity. In diabetes insipidus, the condition is characterized by the inability of the kidneys to concentrate urine due to either insufficient production of antidiuretic hormone, ADH, or a lack of response to ADH. Without adequate ADH, the kidneys cannot reabsorb water properly, leading to the excretion of large volumes of diluted urine, again resulting in a low urine-specific gravity. In summary, a urine-specific gravity less than 1.006 in primary polydipsia and diabetes insipidus reflects the dilution of urine caused by either excessive water intake or impaired water reabsorption by the kidneys. Urine osmolality is usually less than 300 milliosmoles per kilogram. Central diabetes insipidus may be transient if damage is below hypothalamic median eminence or in the posterior pituitary. ADH in hypothalamus can still be secreted systemically via portal capillaries and median eminence. Other words, if someone has central diabetes insipidus, it might be temporary if the damage is in certain areas of the brain, specifically below the hypothalamus or in the back part of the pituitary gland. In these cases, the hormone responsible for regulating water balance, i.e. ADH, can still be released into the body through special blood vessels in the brain, even if there is some damage. The special blood vessels through which antidiuretic hormone, ADH, is released into the body are known as the hypothalamo-hypophyseal tract. This tract serves as the connection between the hypothalamus and the posterior pituitary gland. The axons of magnocellular neurons, where ADH is synthesized, extend through this tract and terminate in the posterior pituitary. When stimulated, these neurons release ADH into the bloodstream, allowing it to reach its target organs, primarily the kidneys, and regulate water balance. This temporary condition occurs because the part of the brain involved in ADH production and release is still able to function to some extent. To assist in understanding, let's revisit the anatomical and physiological process of ADH secretion. Antidiuretic hormone, ADH, also known as vasopressin, is produced and secreted by the hypothalamus and released from the posterior pituitary gland. Here's a breakdown of the process in anatomical and physiological terms. 1. Synthesis in hypothalamus. ADH is synthesized in the cell bodies of magnocellular neurons located in the hypothalamus, specifically in the supraoptic and paraventricular nuclei. 2. Transport to posterior pituitary. Once synthesized, ADH is transported down the axons of these neurons, which extend through the hypothalamo-hypophyseal tract. The axons terminate in the posterior pituitary gland. 3. Storage in posterior pituitary. In the posterior pituitary, ADH is stored in the nerve terminals until a signal triggers its release. 4. Release trigger. Release of ADH is stimulated by various factors, including an increase in blood osmolality, concentration of solutes, or a decrease in blood volume. Osmoreceptors in the hypothalamus detect changes in osmolality, while baroreceptors in blood vessels sense changes in blood volume and pressure. 5. Release into circulation. When these stimuli are detected, nerve impulses cause the release of ADH into the bloodstream from the nerve terminals in the posterior pituitary. 6. Target organ response. ADH acts on the kidneys, specifically on the collecting ducts, to increase water reabsorption. This process helps to concentrate urine, reduce urine volume, and conserve water in the body. In summary, ADH is synthesized in the hypothalamus, transported to the posterior pituitary, and released into the bloodstream in response to changes in blood osmolality or volume. Its primary action is to regulate water balance by influencing water reabsorption in the kidneys.